Namaskar, welcome to PK's classes. Today we are going to study about the leader sequence in TRP operon. So as you know, the operon is a group of functionally related structural genes in prokaryotes. So when we say TRP operon, TRP stands for tryptophan, and we know tryptophan is an amino acid, and tryptophan synthesis is done by this operon. And if you see the structure of the TRP operon, then the genes which are functionally related are 5 in number. So TRPE, TRPD, TRPC, TRPB and TRPA. Then we have the terminator which will stop the gene transcription, and these 5 genes are required for the synthesis of the enzymes which are required for tryptophan synthesis. So if you see the enzymes needed for the tryptophan synthesis, E and D, C, B and A, these 5 genes produce 3 enzymes. So first there will be gene transcription, where we will get the messenger RNA, and from that we will get the proteins, and they are antranilate synthase, then indole, glycerol, phosphate synthase or IgP synthase, and these 2 genes are responsible for production of this enzyme or protein. C is responsible for production of this enzyme, and these 2 genes, TRPB and TRPA, are required for the synthesis of tryptophan synthase. So the tryptophan is synthesized from chorismate. So chorismate to antranilate to phosphoribosyl antranilate to indole glycerol phosphate to tryptophan. So we can say that from chorismate to antranilate, then from antranilate to IgP, indole glycerol phosphate, to tryptophan. So for the synthesis of tryptophan, we need these enzymes, and these enzymes are produced by transcription of the 5 genes TRPE, D, C, B, A, followed by their translation. And there is a regulatory protein called TRPI, which produces the suppressor protein, which we can call this as also TRPR, because it regulates TRPR, which produces the suppressor protein, which is actually inactive. Then we have the promoter, then we have the operator, and before the TRPE gene there is the leader sequence, and today we are going to discuss about this leader sequence. So before that, this regulatory gene TRPR produces the suppressor protein, which actually is inactive, and it becomes active in presence of tryptophan. So when tryptophan is adequately synthesized, it makes this suppressor protein active, and it will block this active suppressor protein will block this operator. And to the promoter, the enzyme RNA polymerase binds. So RNA polymerase is required for the transcription of DNA to messenger RNA. DNA or genes, all the genes will be transcribed to messenger RNA. When RNA polymerase will move along this path, so one by one all the genes will be transcribed, and then there will be translation. So on the messenger RNA, the ribosome will move in this way. So in the messenger RNA, 30S subunit and the 50S subunit will provide the tRNA with the anticodon, and messenger RNA has the codon. Messenger RNA has a codon and the tRNA with a specific amino acid, and the codons of messenger RNA will be recognized by the anticodon of the tRNA. This tRNA will come to the 50th subunit, and there will be supply of amino acid one by one, and there will be synthesis of proteins like these enzymes. So first there will be movement of RNA polymerase, then there will be movement of the ribosomes, and in this way all this protein synthesis occurs. And if this active suppressor protein blocks the operator, suppose blocks here, then the RNA polymerase cannot move, so there will be no transcription or no translation, so it will stop. So in this way, when there is sufficient tryptophan, it will make the suppressor protein active, and this will block the operator site, and there will be no RNA polymerase activity. This is one. So the TRP operon regulation is done by one, this suppressor protein, and second, it is operated by the leader sequence. So we will discuss the leader sequence now. So leader sequence has 4 sections. Leader sequence has 4 sections: 1, 2, 3, and 4. And this 3 has affinity for both 2 as well as 4. So it can bind with 2, 3, or there can be binding of 3 and 4. So the 2-3 binding is weak because there is less number of GC, guanosine and cytosine binding, and when it is 3-4 binding, this is strong, and because there is high number of GC bonds. So 2-3 is weak, whereas 3-4 is binding is strong, and it acts as a termination loop. So RNA polymerase cannot move. Okay, so here, if that means this leader sequence can form two types of bonding, 2,3 as well as 3,4, and when it is 3,4, then if it is 3,4 combination will be here 3,4, then RNA polymerase cannot move and it will stop here. So in this way also it can regulate. So this 3,4 binding acts as a termination loop, whereas 2,3 is weak, so it is called anti-termination loop. So how this is regulated? Now we will discuss. In the one, there are two codons of UGG. That means this UGG is the codon for the amino acid tryptophan. So this tryptophan will be synthesized when this codon in the sequence 1 comes. So when there is low tryptophan, two conditions can be there. One, when there is low tryptophan, and when there is high tryptophan. When there is low tryptophan and when there is high tryptophan. When there is low tryptophan, that means the ribosome. So as we said, first the RNA polymerase will move. And then the ribosome, this ribosome will move, and ribosome is required for the protein synthesis. Ribosome is the site of protein synthesis, and when these codes UGG comes, then the tRNA, the transfer RNA with tryptophan. So suppose here in the messenger RNA there is UGG, then it will be recognized by the anticodon of that tRNA with tryptophan. So the ribosome will wait. When there is low tryptophan, that means the ribosome will wait for that tryptophan. So suppose here in the structure we say, so the RNA polymerase is now say at sequence 4, whereas the ribosome is still at the sequence 1 because there is low tryptophan and it is waiting for the synthesis of tryptophan, because this sequence 1 has codons for tryptophan. So in this condition, what will happen? There will be binding of 2 and 3. So if there is low tryptophan, there will be binding of 2 and 3, which will act as anti-termination loop. So this is because this is weak, and then it will go for tryptophan synthesis. Whereas when there is high tryptophan, it will not wait for these codons in the sequence 1, and so here we can say when it is low tryptophan, 1, 2, 3, 4. So the ribosome is at sequence 1, whereas RNA polymerase is at sequence 4. But when it is high tryptophan, ribosome will follow the RNA polymerase, and it will not wait there. So ribosome is at 1, RNA polymerase is at 2. So 3 and 4 are free. So there will be binding of 3 and 4, which is strong GC bonds and acts as anti-termination loop. So the RNA polymerase cannot move and it will stop the transcription and there will be no synthesis. So in case of low tryptophan, it needs synthesis of tryptophan, and this is the condition how this leader sequence helps. And when there is high tryptophan, there is no requirement of tryptophan synthesis, and this is how the leader sequence by binding of 3 and 4 stops the tryptophan synthesis. So this is all about the leader sequence, how it regulates the TRP operon. Thank you.